Welcome back to Newtonian, and in today's session what we're going to be going through is proteins. Now, what I've started to realize is proteins is one of those chapters which once you learn, you end up applying it in so many different places. So you use the same concept in nuclear chains, so you've got DNA, but you also use the same concept in carbohydrates as well. So I feel proteins is one of those concepts that you really need to draw into. And it's not something that's very difficult. It just requires you to, I guess, have a bit of imagination. Okay, so let's get going. So, what do we actually understand by what a protein is? Now, a protein, like a lot of other things that we'll come across, so a carbohydrate, is essentially a collection of many smaller subunits. So let's have this here, let's draw that here, let's draw that here, and so on, okay? In both directions. Now this is kind of what a protein looks like. So what are these subunits? Now these subunits are something known as amino acids. Now the first thing, what is an amino acid? So an amino acid is the subunit that ends up joining together to end up making a protein, which is a macromolecule. Now, an amino acid itself is made out of several different elements. Now, the elements that amino acids are made out of, they start to make a lot more sense once we look at the actual structure of an amino acid. But still, carbon, hydrogen, oxygen, and nitrogen, these are the most basic elements that every amino acid will always have. Now there are further atoms which amino acids can have, and you don't need to memorize these right now because you'll just kind of intuitively know. And I'll show you how you know those. So one of the most common other elements that is an amino acid is sulfur. Now you might be like, sulfur? Where did that come from? You'll realize in a few minutes. Okay, so the first thing that I want to do here is actually go through the structure of an amino acid before we go away into learning more about proteins. Because if you don't understand what the subunit is, you will never understand what a protein is. You always just think that proteins are just the things that people at the gym have. And that's not necessarily true. We all need proteins in a certain amount, right? So, an amino acid. Structure of an amino acid. How are we going to do this? Now at A level, amino acids are rather simple. At medical school, when you go with amino acids, yeah, things get a bit more different. But at A level, you just need to know a basic amino acid. Okay? So you've got the carbon there. Attached to the carbon, you've got a group here. Now this group, if you're doing A-level chemistry, you will come across a lot, and it's an absolute nightmare. I hated organic when I was doing A-level chemistry, but hey, we've made it past that. So you've got this group here, which is known as your carboxylic group. It's found on one side of your amino acid. Okay? On the other side of your amino acid, you've got a lovely nitrogen, and this nitrogen has two hydrogens attached to it, which is why we'll put NH2, okay? This is your amine group, lovely, okay? Now at the bottom here, we're gonna say we've got a hydrogen, and on top of here is something that's very important. Now what's so important about this one? This one is an R group. Now you'll be like, R? I've never seen an element that looks like an R before. You're not going to see an element that looks like an R, because it's not an element. Now what the R group is something that's very, very important in proteins. R group is almost what gives the protein an identity. It's like the face of the amino acid, right? So we all are humans. What's making us different is our face, right? The structure of our face. As a simplification, right? So the R group is the identity of the amino acid. So this R group could be a whole complex, long structure of elements, or it could be just like something random like a hydrogen maybe, or something sulfur. It can be very complex, it can be very basic. Doesn't matter. It just means it provides the properties to my amino acid. And it's what makes an amino acid different from one amino acid and the other amino acid, okay? R group is a unique structure of elements which makes unique amino acids. Okay, do you understand that? So that's my R group there. I've got my NH2, my amine group here. I've got my carboxylic group there. That's it, okay? That's all the structure there is to an amino acid. Now, we've learned what the structure of an amino acid is. Now, unfortunately, we need to know how two amino acids end up coming together and making two amino acids together, dipeptide. How do they do that? Let's find out, okay? So what we're going to do is we're going to rub all of this off. Let's rub this off. And we are going to draw two lovely amino acids, okay? We don't care about the R group right now. We're going to come to the R group later and why that's important. Right now, we need to figure out how two amino acids come together and make a dipeptide. Ugh, I've always hated chemistry, but life. Carbon here. My R group up here. You know what? Let's just put R group like that. We know R group, what R group is. On this side, we've got NH2, which we said was the amine. Okay, remember amine? Lovely thingy there. At the bottom, we've got a hydrogen here. And we're going to use a different color now. We've got, shall we use a different color? Yeah, we will. Yeah, we've got a carbon here. Double bond oxygen, hydrogen. That looks like a beautiful amino acid. What we're going to do is we're going to call this amino acid 1. Okay? We don't care what amino acid is. All we care is this thingy here. Because we want to figure out how does this join to a fellow amino acid. Let's draw another amino acid. Same thing here. For those people who are pedantic, we can call this R1. Who cares? This is R2. Like R2 just is another R group. Don't care about the R group. I don't care at all about the R group. And on this amino acid, your carboxyl group is a bit irrelevant. Who cares about it? Forget it. Instead, what's relevant on this one is your NH2 group. God, we need to use a different color. Yeah, so we've got nitrogen here. And attached to that, you know what, I'm going to be a bit mean. It's not chemistry, right? Who cares about how bonds look? We're going to draw two. People are going to hate me for it. Okay, let's just put hydrogens here. Okay, so we've got two hydrogens here. That's just NH2, two, fine, easy. And you've got your carboxyl group here. Now, you've got a lovely guy known as an enzyme. Now, we're not going to talk too much about enzymes because you've got a whole university degree ahead of you to meet lovely enzymes. So who cares about them now? Enjoy not knowing about enzymes. Okay, you've got a certain enzyme that just rocks up and he's like, amino acid 1, amino acid 2. Today is the day you react together and we'll make a dipeptide. I can't make this any more exciting, can I? Okay, so this oxygen here, this hydrogen here, they're going to be part of the reaction. What else is going to be part of the reaction? This dude here, this other hydrogen there. So you've got the OH, the oxygen, and the hydrogen from one amino acid will be reacting with the hydrogen of the amine group of my other amino acid. How fun. Now, some of you might just realize there's a pattern here. Let's see how many of you figured that out. Now, if these are reacting together, what are the elements here? One hydrogen, two hydrogens, H2. H2. Oh, there's an oxygen as well. Oh my god, have I seen H2O before? Yeah, it's water, guys. Life is simple. Well, at least I'm trying to make it simple. So that's water. So when amino acid 1 reacts with amino acid 2, you're forming water. What did we call water? Being formed, water forming reactions in the carbohydrate video? Condensation reactions, right? So this is literally just a condensation reaction here. Condensation reaction. You see, this is one of the good things, right? With like carbohydrates and proteins and stuff, like some of the terms are literally just the same. It's like, yeah, fine, okay. Now, what we need to do is realize what happens to this structure now. I know that I formed a water molecule, but we're not trying to form water here, right? We're not trying to make it rain or anything. What we're trying to make is a dipeptide having this amino acid joined with that amino acid. So let's figure out what the structure is going to be. So the way most people will do this is literally just draw how it looks. And then there's me sitting there and I'm like, how did we get there? So what we're going to do is something a bit better. Which is basically going to be like, yeah, you'll see. Because this is gone, forget it. Who cares about them? They're water. They can evaporate somewhere. I don't care, right? But what do we have left? Well, we've got a few things left. We've got this carbon, we've got this oxygen, we've got this nitrogen, and we've got this hydrogen. Now, as life is, the hydrogen's always ignored for some reason, right? So who cares about that hydrogen? The hydrogen is there. It's there, right? What we care about is the carbon and the nitrogen, and probably the oxygen too. So what do? We're going to redraw it. So we know these guys are irrelevant. Rub them off. That one doesn't exist. Nor does that one exist anymore. Lovely. Now, what bond can exist? Well, because we got rid of these, this carbon's like, oh my god, I need to make a bond with someone. This nitrogen's like, yeah, I need to make a bond with someone as well. You've taken away my hydrogen, not that I cared about him, but hey. So they realised the only place that they can actually make a bond is between the nitrogen here and the carbon there. Yeah, very creative. But yeah, so literally, boom, you have a bond between them. And this bond is known as a peptide bond. Yeah, there's nothing more to it. Not sure how I make this more exciting. But yeah, that's literally a peptide bond. Okay. Yeah. Okay. So we figured out what a condensation reaction was and how we form a peptide bond, but wait, what's the other way around? What if, I'm like, we need to break the protein down. We need to break the dipeptide down. We're feeling very destructive today. What do we do? Well, the first thing is we have an enzyme that rocks up. As always, enzymes are needed. They're too important. They're like celebrities. They make everything work. And you need the thing you lost, which was a water molecule. So the water molecule decides to turn up. He's like, hey, let's do something. The enzyme's like, yes, we definitely need to do something. So the enzyme facilitates the water to go back. The water sits jolly way back here, and the enzyme is doing its enzyme-y thing, and it decides to break the bond. And when it does decide to break the bond, what you realise is things need to go back to normal, because there's no bond here. The carbon's like, oh my god, I need to make a bond with someone again. And you realise that that H2O kind of looks like this, right? And the carbon's like, hang on a minute, I remember you, OH. You were from me. So, my guy, the carbon, decides to take the OH back, and you have your OH returning here. OH. Lovely, you've got amino acid 1 reformed, amino acid 1's happy. Now the nitrogen's like, okay, he's decided, the carbon's decided to abandon me, so I kind of need to make another bond. So what the nitrogen decides to do is take this hydrogen back, and it forms NH2, and this bond goes into oblivion, and these two amino acids are reformed as themselves, and we're back to the start. Now, the one more thing that we need to know, is what type of reaction is that? Is it still a condensation reaction? Are we producing water? No. What are we doing? Are we giving water into the reaction? Yeah. What did we call that before? We called it a hydrolysis reaction. So it's no longer a condensation reaction. Please don't be the silly person who writes it's a condensation reaction, because it's not a condensation reaction. It's a hydrolysis. Because hydro means water, lysis means breaking things down. Can't lie, scientists probably don't get any more creative than this. Oh, no, they do. Wait till you get to med school and you learn names of conditions you can hardly pronounce. Yeah, not fun. So, okay, so we've done all of this. Cool. Let's move on to the next thing. Now that you know these subunits and blah blah blah, the amino acid chemistry, we need to figure out what in the world is a protein. And the thing is, right, if you look at a lot of resources, what they will now decide to do is they will decide to tell you the structure of a random example protein. And then you scratch your head and you're like, what in the world is this? Or at least I did. So how about we learn the basics? And once we know the basics, we can learn anything, I guess. Yeah, okay. So the basic number one. What are the different levels of structure in a protein? Well, there are 4 levels of structure in a protein that you need to remember, and you will always need to remember. Like, it just turns up everywhere. My first year was literally filled with doing this. So, first structure of a protein, the most basic structure. What do you think we're going to call it? It's the most creative thing. We call it primary structure. Oh my god. Primary structure. Primary structure of a protein. Lovely. Number 2. Yeah, secondary. Secondary structure. Number 3. I'm sure you guessed it. It's tertiary. Number 4. Believe it or not, it's quaternary. God, I love this. Okay. Now I've written them all out, we need to figure out what in the world is each one of them. And that's kind of, you know what, that's where a lot of students actually get confused for some reason. Maybe because they're not told well enough what in the world this is. Or they think like, oh my god, it sounds too sciencey. Nah. Okay, let's go through primary structure first. What is primary structure? Now, if you guys did your GCSEs, or at least decided to try and pay a bit of attention at GCSE, you know that you've got DNA, right? Or I hope you would have known that, unless you were kind of living under a rock. But yeah, the DNA, so this thingy, A, T, C, G, G, A, blah, 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 whatever you want to call it. So these are the different codons. You probably don't know what codon means, but hey, it's just one of these DNA sequence, guys. Just know it like that. You'll learn it later on. I think you learn it at GCSE. Can't even remember anymore. But yeah, so you've got this whole code. Now, what you need to know is in your DNA code, each three bases are kind of important. Why? Because they code for an amino acid. So each 3 of these ones code for an amino acid. So this codes for amino acid 1, this codes for amino acid 2, and so on, and so on, and so on. Now in total, your body, funnily enough, only has 20 amino acids. That's not a lot if you think about it, but you know, you'll actually cover out why 20 is a bit of a weird one, because technically I think you can have 64? Maths might be out. But mathematically you should be able to have a lot more than these numbers. But then you realize there's degeneracy of code, blah, 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 genetic stuff. That causes you only to have 20. You don't need to know that right now. You will learn that later on. But for now, remember, every 3 bases equal 1 amino acid out of the 20. Easy as that. Now, the order of these 3s will of course tell me what the amino acids are, right? So if this is like amino acid 1, we'll have amino acid 1 rocking up, and then joining to amino acid 2, which will then join to 3, and blah blah blah. It's like an instruction code, for God's sake, right? That's what your primary structure is. Primary structure, in basic words, is the order of amino acids in a protein which is determined by the DNA sequence says codons. Okay, codon means 3 bases, okay, which equal to an amino acid. Those amino acids join together in order. We form primary structure. Yeah, that's all the primary structure is there. So primary structure will end up forming long chains of these amino acids, which kind of is what a protein kind of looks like, but in a more complex structure. So that's your primary structure. Yeah, primary structure is kind of an easy one, to be honest. Next comes the bit more fun things. Secondary structure. Now, secondary structure, I can't lie, all of these structures are easy. Like, it's just like you need to wrap your head around them once and then you're set. I can't lie. With A-level, it's just like, if you put enough effort, they're not even that bad. I lied, the exam questions are much harder, the content's calm. Okay, yeah, I digress. Secondary structure. Secondary. Now, secondary structure, you can have two structures basically in secondary structure. The first one that you can have is something known as an alpha helix, okay? Now, we don't need to go into much depth with the alpha helix, or the next thing you'll learn about it at maybe medical school, biomed, or you will never learn about it and you can live a happy life. Yeah, and the second type is a beta pleated sheet. Yeah, alpha helix and beta pleated sheet. Now these are the only 2 types of secondary structure that you're going to come across at A-levels, okay? Just imagine that's it. Secondary structure, either out of either this one or that one, okay? That's it, right? Now, how does an alpha helix look? Well, it's called an helix for a reason, for God's sake, right? It's just like, kind of like that. Now, a few important things about alpha helix. What's the bonding in them, okay? How do we form that structure, and what's the most common coiling? Now, the most common, so essentially a helix is like twisting around, blah, 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 2 strands twisting around one another. Now, most of these strands are always right-handed. Now, this is because it's the most stable configuration. You don't need to know what stable means, don't bother looking it up. You don't need to know, I barely understand it, well, I kind of do, but don't look it up, right? Right-handed. I can't spell, can I? Right-handed, yeah. Now, most are right-handed. Notable exception is collagen, if I remember correctly. I think, yeah, it's collagen. Collagen is left-handed because it wanted to be different. Don't ask me why. We don't need to know why. It's just left-handed is collagen, but mostly it is right-handed. Right-handed helix. Alpha helix is mainly right-handed. Just remember that. Please don't question it. And collagen is a notable exception, but yeah. Now, in both alpha helix and beta pleated sheets, there's only one type of bond present. Only one type of bond. Remember, please don't mix it up. People mix it up so hard that I'm like, oh, don't do it, right? The only type of bond in secondary structure, i.e. in a alpha helix or a beta pleated sheet, for God's sake, is just a hydrogen bond. Hydrogen bond, please don't mix it up. Hydrogen bond. If I could like come into the camera and like just knock on your head and say it's hydrogen bond, please remember, right? Now this forms between what? Do you remember the R groups I was talking about? Yeah, basically you'll have R group 1 rocking up and R group 2 rocking up. And because well, I probably should have told you this. No, I hope you guys have worked this out. So if you've got your nitrogen here, you've got hydrogen here, you've got your carbon here, you've got your oxygen here, you've got the oxygen here, you've got OH here. Now, what you'll realize is that these are partially positive and partially negative. Is that too much enthusiasm for this? It probably is. Probably looking like a weirdo, but hey. So, because these are partially charges, the partial charges are different, you'll have attraction, right? But now you need to remember that these are not this is not the end of an amino acid. These are your R groups, okay? These are your R groups, not the amino acid's end, okay? So your amino acid will be here, like for example, you'll have your NH2 here, you have your carboxylic group here, okay? I hope you see it. So that's your carboxylic group and amine group there. That's your hydrogen here. If you just rotate it all around, okay? Now this is your R group here, and this is the R group for the other amino acid. Right, I hope that kind of, you can see it. And this is the attraction you'll have, okay? Now the attraction between these two is a hydrogen bond, right? The hydrogen bonds between the R groups are what causes secondary structure to occur. The two types of secondary structure there is a alpha helix and a beta pleated sheet. Alpha helices are most commonly rotated, have a right-handed rotation. Common exception, well, notable exception is collagen, which is left-handed. Alpha helices. Now, this is something you just need to remember what I wrote, okay? Every fourth amino acid, sorry. I am saying this right? Just make sure I am. Yeah. So every fourth amino acid in the protein will have hydrogen bonds which cause for the coiling to occur, and thus alpha helix to form. That's all you need to know about alpha helices. Alpha helices have hydrogen bonds between every fourth amino acid. They basically make a twisting structure. Don't even try to figure out why it's the fourth one. It just is, right? This is not what's going to get you your A star. This is something you need to know. It's a fact you need to know. And that's fine. You learn it and you move on, okay? You don't spend stupid amounts of time trying to figure out niche stuff, because that's not what A-level is about. A-level is learning the content, trying to figure out how in the world I apply the content, right? Critical reasoning is very important. Yeah, sure, you need to know the content as well, but please, critical reasoning. Okay, that's alpha helix done. Let's go over a beta pleated sheet, which is the second type of secondary structure, okay? Beta plated sheet. Here. Lovely. How does beta pleated sheet look? Now, if I wanted to complicate your life, I could draw a lovely complex structure, which you don't need to know. But I won't, so I'll just draw a simplified structure. So those of you who looked this up, please don't criticize me for this. Yeah, don't bother. Not that it matters. But essentially, you'll have rows of amino acids. Okay, rows of amino acids. These amino. Let's draw in 3D. How about that? Right, the rows of amino acids. No, that'll be confusing for you guys, wouldn't it? Yeah. Okay. Fine. You'll have rows of your amino acids. Rows of amino acids. Essentially, between them, you'll have bonding occurring. Now, if you notice, I've drawn these bonds, these hydrogen bonds, in a very specific way, because the way I've drawn it is it's at angles. So what this actually causes the beta pleated sheet to kind of look like is this. If you guys can kind of figure out what my... I thought DT was meant to help me be good at drawing in 3D, but hey. If you can see, that's kind of like a 3D structure. Can you see it? It's like a beta pleated sheet. Yeah, please. I hope you see it. Now these are your chains. Your chains, you know what? I probably should tell you. So the most one type of beta pleated sheet is kind of like this. Okay. Now this is your protein chain. Okay, these protein chains will fold over. Blah blah blah. This is amino acids. Here, all amino acids in your protein chain. Amino acid, amino acid, amino acid, amino acid, amino acid, amino acid, amino acid. Now these amino acids, where's a different color? These amino acids have hydrogen bonds. Hydrogen bonds. All of these are hydrogen bonds. These hydrogen bonds are causing, well, beta pleated sheets to be made. Again, the same rule. Every fourth amino acid has a hydrogen bond with the other fourth amino acid, right? So 4, 8, 12, blah, blah, blah. They have hydrogen bonds between each other. And the R group is where the hydrogen bonds are occurring between. Please remember this. Don't go and tell me that the amine group at the end of the amino acid is having a hydrogen bond with the carboxylic group of another amino acid. Like, just don't lose your mark in a better way, right? That's a beta pleated sheet. Done and dusted. Don't need to know. I don't think you need to know anything else about this. Yeah, that's it. You're done. You're done with beta pleated sheets. I don't know why people complicate these so much unnecessarily. Like, yeah. What are we gonna learn next? Well, we've done secondary structure. We're an ASAT secondary structure now. Well, we need to become an ASAT. Well, the next one, don't we? The next one, what's the next one? Take a guess, Sherlock. It's tertiary. Tertiary structure. Now, this is where things get a bit more interesting and fun. Because you need to know a bit more. Again, tertiary structure between our groups. Don't be the silly one saying something else. Between our groups. Now, with tertiary structure, and you'll later realize quaternary, sorry, they've got a specific type of bonds which, well, form the tertiary structure. Yeah. Well, okay. What are they? Number one. You've got hydrogen bonds. Who could have guessed? Number 2. Disulfide bonds. You know what? I think I should improve my spellings. Disulfide. Number 3. Hydrophobic and hydrophilic interactions. Yeah, you've seen this before, haven't you? Yeah, it's the same thing. And finally, oh God, you've got my favorite. Don't know why they're my favorite. They just sound cool. Ionic bonds. Let's quickly go through all of these bonds. Why does a hydrogen bond occur? A hydrogen bond occurs because there's something that's partially positive and there's something that's partially negative. Partially positive decides to attract partially negative. Ooh. You've got a bond, and which is called a hydrogen bond. So an R group that's partially positive will attract an R group that's partially negative. Sparks fly. Disulfide bonds. What are they? But what does di mean? No, not death, right? No. I'm talking about die in, like, I don't even know what language it is, but di means 2, okay? Right, the next thing people say. 2. What does sulfide mean? Well, sulfide means something to do with sulfur. 2 sulfurs. I don't know why people don't see these things in such a simple way. It's 2 sulfurs, for God's sake. You've got one R group that has a sulfur, you've got another R group that has another sulfur. That's it. So, the R group 1, the sulfur, he's like, dude, I need to bond with someone. And R group 2 has another sulfur, he's like, dude, I need to bond with someone, I need to hang out. So they both decide to form a bond between each other, and that's called a disulfide bond. Yeah. Hydrophobic and hydrophilic interactions. Hydrophobic, hydro means water, phobic means oh my god, I hate this. That means hydrophobic means something that hates water, right? So if you've got an R group that hates water, it will of course be friends with an R group that also hates water. Yeah? Like similar fashions, I don't know. So they decide to hang out and bond with each other. So hydrophobic group, R group, hydrophobic R group, join together, boom, we've got a bond forming. And then you've got hydrophilic. What does hydro mean? Water. Philic means we like each other. So they like water. Amino acids which have R groups that like water decide to stick together. They decide to hang out with each other, chill out with each other, and form bonds. So you've got another bond forming. Ionic bonds, my personal favorite, have a soft spot for them. Well, they're formed because of different charges. So if I've got something that's negatively charged, okay, please don't confuse it with a hydrogen bond. Hydrogen bond is a partial charge, right? This is a full charge, right? It's like it's the real deal, right? So you've got, God, my voice is gonna go. You've got a negative charge here, and you've got a positive charge here. What's gonna happen? Come on, Sherlock. They're going to attract. How do you draw attract? That thing? Yeah, they're going to attract, right? You've got another bond formed. So, what are the 4 bonds that you can have that make up tertiary structure? First one, hydrogen bond. Second one, disulfide bond. Third one, come on. Or the third one is hydrophobic and hydrophilic interactions. The fourth one is an ionic bond. Now, these all, between R groups, happen and you end up forming cool structures. And you don't need to know a single name of these structures. Well, not at least to A-level, please don't. And what you actually end up having is these alpha helices, these beta pelated sheets, they all end up like contorting around, blah, 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 and they end up making something that kind of looks like this. You guys are probably laughing, what is he drawing? That's kind of how a protein actually looks, right? It's not like that, or I don't know, how do you imagine a protein? I've never done it myself, but that's how a protein looks. And that's because it's got like, it's got so many different bonds. You know, I remember being in an A-level bio lesson and my teacher drawing that and then putting an S there and putting an S there, and then my friend being like, is that a disulfide bond, miss? And I was like, how did you know that? It was only later I realized the only reason he was saying is that a disulfide bond here is because there's 2 sulfurs. There's 2 sulfurs there. And between 2 sulfurs, what does 2 mean? It means di, and then you've got 2 sulfurs, disulfur, disulfide. It's a disulfide bond. I was just a bit slow to understand there. But hey, you get it, right? So these different 4 different bonds, they cause between the R groups of amino acids, they cause the amino acids, the actual structure of the protein to like contort even more. I'm not a contortionist, so I'm sorry. But they cause a very cool 3D, please remember 3D, I can't draw 3D, unfortunately, but form a 3D structure, which is very complex and looks crazy. It probably is crazy, but yeah, it's a 3D structure which is formed, and that's a tertiary structure. Now, how do we remember these 4 bonds? Like, you saw me, I kind of knew them off by heart, but how do I know them off by heart? I hate learning stuff off by heart, so how do I do it? Well, it's easy. The way I want you guys to remember it is, well, you can remember it, probably, is through this mnemonic. HD, hi, right? So you're like, it's Sky TV is HD, and you're like, hi. I don't know if that makes it. Or you can like try and make remember it through the phonetic, like hud, hi, hud, hi, hud, he, whatever, or HD, hi. Yeah, you get the point, right? It's a mnemonic. Remember it. Maybe else, maybe doesn't. Who cares? It's fine. We move on. We move on to quaternary structure. Why am I honestly this passionate about this? Trust me, I was not this passionate whilst I was learning this for an exam. Okay, final type of structure. Quaternary structure, number 4. We get to finish this quickly and you get to turn off the video and then, I don't know, enjoy life. Quaternary structure. Lovely. What are we going to do here? Now, quaternary structure. Why did I even rub what I... You'll see what I mean. Right. Quaternary structure. We've got 4 bonds. Let's be creative. What could they be? You've got a hydrogen bond. Number 2, disulfide. Number 3, why am I having to write this out again? Hydrophilic and hydrophobic. And number 4, our lovely ionic. Now, you're like, what? You've just drawn the same thing out. What are you even doing? Well, quaternary structure and tertiary structure use the same bonds. They use the same bonds. Yeah. Makes me want to cry, having to write all that out again. But they use the same bonds. But what's the difference? The difference is this is no longer to do with just one amino acid. Sorry, one peptide chain. Quaternary structure is interesting because quaternary structure allows us to essentially combine several different long chains of amino acids which are not otherwise joined together. So say I've got chain 1 here, right? And I've got chain 2 here, right? These are 2 separate chains, and they're in reality like contorted like ooh, like that kind of, and then that's ooh, like that. But these 2 want to have some bonds with each other, want to form a more cool structure, right? They want to form this, let's say. I've just broken the chalk. I am so good. Oh God. Right. Okay, so they want to form that. How are they going to do that? All they do is they employ these bonds. They might form a hydrogen bond here. They might form a disulfide bond here because there's 2 sulfurs here. They might form an ionic bond here because there's like a positive here, negative here. They might form another bond, a hydrophobic or hydrophilic bond here. Essentially, they'll form bonds between different long chains of amino acids between their R groups. So they don't actually have peptide bonds between each other. So amino acid chain 1, the chain 1 does not have any peptide bonds with chain 2. But instead, they've got these 4 different types of bonds between each other. That's it. That's what quaternary structure is. You know, I've seen too many teachers and too many textbooks like try and explain it in like some long-winded method of like, it's just simple, right? Keep things simple. Once you've covered the basics, you can always learn more. What you need to know is the basics. That's what's going to get you your A star, right? Knowing the basics and having those solid and then building up on them. Not going into silly depths which no one understands and then like no one knows anything. That's not why we're here, right? Okay, so now let's go on to the next. Okay, so now what we're going to do is we're going to go over a very key and fundamental concept, okay? So, yeah, okay, fine, let's go about it like this, right? So what are the 2 types of proteins that you can have? The first type of protein you can have is a globular protein, okay? The second type of protein you can have is a fibrous protein. Now, you know, most people will just tell you definitions, tell you to memorize them. I'm not going to tell you that right now. I will eventually, but not right now. What I want you to do is look at the words. What does globular mean? Well, there's 2 ways of looking at it, right? The way I saw it was like globular, I don't know, it reminded me of like a blob, right? Glob, blob, glob, blob. Rhymes? Yeah. Right, so reminds you of a blob, like a circular blob or something. Or you can be a bit more pedantic about it, and you can realize that glob and globe sound a bit similar. And that just means like a sphere, right? Essentially like a sphere. That's how this protein kind of looks like. Who knows what that means? It's just a globular protein. A protein that looks like a globe is how I remember. Okay, that's what a globular protein is. What about a fibrous protein? Do you want to take a guess? Well, when you're eating chicken, I'm a vegetarian, probably not the best at this, but when you're ripping apart the meat, you'll notice it's very fibrous, and you'll call it like very fibrous because what does fibrous mean? A lot of fibers, right? Those small stringy things are fibers, and that's what fibrous means. Fibrous is essentially a lot of fibers, right? Or a lot of repetition, right? So fibrous proteins are proteins which have a lot of repetition and are in like long chains and don't look like a globe. Yeah. Oh, I've chucked the chalk. I'm gonna have to get it later. No more orange color for us. Okay. Cool. Let's learn about globular proteins first, because I think globular proteins are where you need to know a bit of a fundamental concept. So let's go through these first. So at A level, you need to know is how do we form a globular protein? And you're like, oh my god, I need to know that. Yeah, it's easy. It's really calm. Now, how are we going to do this? Now, the way to learn how a globular protein is this. You need to have understanding behind it. Now, we know that there's two types of water loving or water hating, right? It's either one that in our group can be. Now, a long amino acid chain, it will have many amino acids, okay? With many different R groups. Many different R groups. And some of these will be hydrophobic and some of these will be hydrophilic, okay? Now, these globular proteins are formed in water, in an aqueous environment. So this all is happening underwater, like in a cell, which is aqueous, which is water. I hope that was self-explanatory. But it's forming in water, it's forming in an aqueous environment. This means that the water-hating guys, the hydrophobic amino acid R groups, they're like, God, the water stinks. We don't want to do anything with the water. And what they decide to do is they come up with a core plan. And then they talk to the hydrophilic amino acid R groups and they're like, you love water, don't you? How about you go and meet water and be with water, be one with water? And the hydrophilic guys are like, yes! They become fully passionate. What actually ends up happening is the hydrophilic guys end up all being at the outer edge. So, how are we going to do this? Well, okay. First, you've got water everywhere. And wait, bear with me. I need to get my orange. Now, this orange guy will represent my hydrophilic amino acid R groups. These hydrophilic amino acid R groups line up on the outside of the globular protein. And this is because they're able to interact and be one with the water. They can be happy with the water, having a fun time. The hydrophobic guys are like, ugh, we don't like water. So they decide to line the inside of the globular protein. And this means that you end up with a globular protein which is hydrophilic on the outside but has a hydrophobic core. So hydrophilic outside and a hydrophobic core. Okay, so the inside is hydrophobic and the outside is hydrophilic. Now, why is this important and why is this useful in bio? If you've got something that's water-loving on the outside, this means that this protein is soluble in an aqueous environment. That means it can be carried in aqueous environments, like inside of a cell, or, for example, in the bloodstream. Right? That's essentially all a globular protein kind of is. Okay, so now let's go through some examples of globular proteins you need to know at A level. Okay, what's the first one? Well, the first type of globular protein you need to know is insulin. Second type, hemoglobin. Third, catalase. Fourth, amylase. Okay, now what you realize, this guy is essentially a hormone which is released by beta pancreatic cells in the pancreas, and essentially is to do with glucose levels and maintaining them. Now this is released into the bloodstream, okay? Hemoglobin is also in blood, and catalase is in aqueous environments of the cell. cytoplasm, sorry. Same thing here. Okay? Catalase and amylase, aqueous cell cytoplasm. Okay? Insulin and hemoglobin are in the blood. Oh my god. Yeah, this is actually tiring. But okay, I digress. Insulin, why do we want insulin to have a globular protein structure? Think about it. Yeah, it needs to be aqueous. If the outside was hydrophobic, do you think insulin would be happy going in the bloodstream? Oh, do I want to go there? No. But because the outside of insulin is hydrophilic, because it's a globular protein, insulin is like, let's go into the blood. I wonder if insulin does that, but I imagine it, right? Okay, so insulin is a globular protein because it needs to be aqueous in blood, and once it's aqueous in blood, life is happy. Insulin is in blood, it does what it needs to do. Yeah, that's it. You don't need to know the structure for insulin. Don't bother, please. Hemoglobin, you do need to know the structure. Welcome to the land of hemoglobin structures. They're dead easy, trust me. Now, people will give you random long diagrams of hemoglobin that look nothing like hemoglobin either, but just look a bit more sciencey. Don't bother with them. Learn the block model, or at least that's why I like calling it. So hemoglobin is kind of like this. Okay, alpha, and because scientists are exceptionally creatively minded people, they decide to come up with creative names. Oh, they just decided to go with alpha beta again. Yeah, so that's kind of how hemoglobin looks. Now, what's interesting with hemoglobin and catalase and amylase, these are proteins which are called conjugated proteins. Conjugated. Conjugated means together. That means together with what? Conjugated proteins are proteins which have something stuck on them. And this is something known as a prosthetic group. A prosthetic group. Conjugated proteins like hemoglobin, amylose, and catalase have prosthetic groups. Prosthetic groups are essentially like a dude that comes along and he's like, you know what, protein, I'm gonna hang out with you. And this is something like, for example, heme, which is iron, so ions. It could be lipids, or it even could be carbs. Okay, it's like, dude, to the protein, you're so cool, I'm gonna hang out with you. So the Fe, or like the ion, the lipid, or the carb, they're like, let's go, and they join up with the protein. And that's why they're called conjugated proteins, because they have prosthetic groups which could be ions, lipids, or carbs. Now, hemoglobin also does have a prosthetic, it does have prosthetic groups. Now, these prosthetic groups are known as heme groups. Heme, so you've got prosthetic groups here. And these contain iron, okay? Sorry, I'm not gonna die. So you've got the prosthetic groups on your hemoglobin. Now, do you need to know any more about hemoglobin? Let me check. I don't want to give you guys like silly amounts of information about hemoglobin. No, you don't need to know about basically the structure of hemoglobin can change when oxygen joins onto it, and that helps it actually take up the oxygen. You don't need to know it right now, forget it. Who cares? Right? That's the structure of hemoglobin, remember. 2 alpha chains, 2 beta chains, they have they have prosthetic groups. And this is quaternary structure. What does quaternary structure include? It includes hydrogen bonds, it includes disulfide bonds, it includes hydrophilic and hydrophobic bonds, and it finally includes ionic bonds. So you probably have a mixture of that going on here, and who cares about it? I don't. Right? Now, what are we doing? Well, we're going on to catalase. Catalase is an enzyme. Who cares about what it does? Do we care about what it does? Yeah. Well, yeah. I think you actually do need to know what catalase does. Yeah, catalase, yeah, you probably do. Yeah, catalase, yes you do, sorry. Catalase breaks down hydrogen peroxide. Hydrogen peroxide is like, ooh, it's very bad. It's like it's an evil dude. It's like Doofenshmirtz, right? The catalase breaks down hydrogen peroxide because hydrogen peroxide is evil. Hydrogen peroxide evil to body. Body hydrogen peroxide don't mix well. Right. So catalase comes along, rocks along, he's like, oh my god, let's break you apart. Breaks hydrogen peroxide into something a bit more nicer, not that evil, and woo, happy days. Right, now, what do you need to know? Catalase has a quaternary structure and has the prosthetic group. Catalase has prosthetic groups and, in other words, is a conjugated protein. And catalase has a quaternary structure. It's made out of 4 polypeptide chains. What does polypeptide mean? Lots of amino acids, lots of peptide bonds. Woo. Now 4 different chains. 4 different chains. Much like the previous one, hemoglobin, these have bonds between each other. HD high, that was the acronym for it. And they also have prosthetic group. What's a prosthetic group in catalase? Weirdly enough, it's again heme. So heme wants to be everywhere. Catalase has heme as well. Remember it, don't question it. Life is easy. Next one. Amylase. Sorry, I lied. Amylase does not have a quaternary structure. Amylase instead only has a tertiary. So it's only made out of 1 amino acid chain. What does amylase do? Come on, you know what amylase does. You do, right? Breaks down starch. I hope that's what it does. So if it doesn't, yeah. Okay, amylase breaks down starch, has a tertiary structure, has HD high bonds between them. I hope you remember what that means. Now, these different proteins have specific 3D structures which allow them to do their jobs, right? Like, you wouldn't be using a screwdriver instead of a spanner, would you? Or you wouldn't use a spanner instead of a screwdriver to, like, screw a screw, because they've got a specific job. A spanner looks different to a screwdriver because the shape is suited to a certain job, so on. Same way, proteins have to have a specific structure for them to be able to do their job. You don't need to know most of them, but those 3 you did need to know, and that was it. That's it. That's globular proteins done. Remember that, and you're set with globular proteins. What's next? Fibrous proteins. Yeah, those are a bit more annoying, I can't lie. Okay, let's go over those now. Okay, so now let's go through fibrous proteins. Now, what exactly is a fibrous protein? Now, fibrous proteins are essentially long, insoluble proteins which are generally used for structural purposes. Okay? Now, these fibrous, let's use orange here, fibrous proteins. Now, these generally are insoluble, and I'll tell you why. They're generally insoluble, and they generally have hydrophobic R groups. Their R groups are generally very short R groups. The pattern of amino acids, so amino acid 1, amino acid 2, amino acid 3, is very repetitive. So this one will repeat again, 1, 2, 3, 1, 2, 3, so on. So the actual amino acids that are being used, so the R groups, they're very repetitive. So you might have 1, 2, 3, 4, and then again 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, so on. So fibrous proteins have a very repetitive structure in terms of their R groups and amino acids, and the R groups themselves are very short, and most of them are hydrophobic, a very high proportion, and this means they're mostly insoluble. Now, having very short R groups and having them in a very repetitive structure means that they're essentially like they're kind of like the army. They're very like uniform, and they're like strong, right? So that means that they're very strong and they can be used for structural purposes, okay, because they're very uniform. You don't need to know why and more about it, but that's it. Okay, that's the main property of fibrous proteins. Now, there's three main fibrous proteins you need to remember. Please remember them. They're going to come up, and if you lose the mark, I'm sorry. You could have saved it. You could have got that A star instead of the A if you just decided to do this. Collagen. Number 2, keratin. Number 3, don't worry, it's the last thing we're going to do, right? Number 3, come on, try and remember this. I'm probably speaking to myself. Yeah, elastin. Oh God, how did we figure? Elastin. Okay, so realistically speaking, which ones have you come across already in life? Well, the first two, you probably have come across these at some point, collagen and keratin. Collagen you've probably come across like with those skin serums, which are like, we are packed with collagen, which is going to make your skin beautiful, right? That thingy. And then you've got keratin. Keratin is like those hair adverts are like, we are packed with nutritional supplements of keratin, blah, blah, blah. So you kind of realize that collagen is something to do with the skin or tissues and tendons, whereas keratin is something to do with the hair. Elastin is an easy one. Elastin, elastic, elastin sounds the same, right? It is the same, for God's sake. Elastin is a protein that makes things elastic. Simple. Made you learn stuff very quick. But we need to go through these structures. We need to go through the structures of these proteins, and very briefly, not much. More details you can come across through exam questions, but generally speaking, you don't need to know too much. Collagen is made up of a helix. Okay, it's a very helical structure. Now, collagen is a left-handed helix. Left-handed helix. So it basically rotates in a left hand. Collagen is made up of 3 polypeptide chains. I'm pretty sure it is. Yeah, 3 polypeptide chains. Now, these 3 polypeptide chains make up collagen when they are left-handedly helixed or rotated together. The functions of this is for the skin, tendons, and tissue, and it serves as a structural protein for these, right? It's like a rope which is holding things together. It's the rope and the scaffolding. The next one we're going to go through is keratin. Now, keratin is very interesting. Keratin is not only found in hair, it's also found in nails. Yeah. Now, why keratin is interesting, and I like to stress a bit upon keratin, is because it uses disulfide bonds. Now, with keratin, the more disulfide bonds there are between the R groups, the harder or the less flexible the material that contains them. So nails have a high content of keratin in them, which is why when you get those keratin supplement advertisements, they're like hair and nail and toenail supplement, because they all contain keratin, which contains disulfide bonds. And the more disulfide bonds you have, the more brittle and less flexible the thing is, the more harder it is. That's why nails are quite hard, because they've got a lot of disulfide bonds. Whereas when you've got low disulfide bonds, you've got higher flexibility, and stuff like hair is formed, which is like, can you tell it's malleable? It's quite flexible, right? So that's what keratin is all about. Now, do you need to know any more structure than this much? Let me see. No, so that's all keratin is. But the last thing that we need to go over is elastin. Elastin, what I said was it's about elasticity and it provides flexibility. Where is elastin found the most? It's found in alveoli, so things can stretch. And finally, it's also found in blood vessels to help them stretch as well. Do you need to know the structure about these really? No. All you need to remember, elastin is a fibrous protein, and that's it. So these are the 3 fibrous proteins you need to know: collagen, keratin, and elastin. Cool. So don't even know how long this video is going to be, but that's all you need to know about proteins. Proteins have 4 different stages of structure: primary, secondary, tertiary, quaternary. They have primary, that's peptide bonds between the amino acids, and it's determined by the nucleic sequence of the codons, like so 3 bases. Secondary structure is either alpha helix or beta pleated sheets, and those are formed because of hydrogen bonds. Tertiary structure is the random complicated contorted structure of only one chain of amino acids, and it contains hydrogen, hydrophilic bonds, hydrophobic bonds, disulfide bonds, and ionic bonds, okay, which you can remember by HD, HI, or HD high, or HUD high, or whatever, right? Then finally you have quaternary structure. Quaternary structure is essentially several different amino acid chains coming together, and they have the HD high bonds between each other. I won't go through them again, but that's it. And then you've got fibrous proteins and globular proteins. Globular proteins are soluble because they have the hydrophilic amino acid R groups on the outside and the hydrophobic amino acid R groups as the core inside. Fibrous proteins are structural proteins. These are made out of highly repetitive sequences of amino acids, i.e. their R groups, very repetitive. The R groups are quite short, provides it more structural stability so that you can form structural proteins such as collagen, keratin, and elastin. Collagen is found in tendons, tissues, skin, etc. Whereas then you've got keratin. Keratin is found in nails and hair, and that's a structural protein there which has disulfide bonds, right? It has a quaternary structure, and these disulfide bonds, the more they are, the harder the substance. Then finally you have elastin. Elastin is what provides flexibility and elasticity to stuff like alveoli or blood vessels. And that's all you needed to know about proteins. I hope you found this video useful and interesting, and please keep learning at Newtonian. Thank you.